Cool. Okay, so welcome to HPL seminar for the next hour and a bit. I'm Ashnik Ambari. And today we are gonna Dr. Steph Roth is gonna be presenting for us. Steph's educational background, she did her Bachelor of Science from SFU in British Columbia in the Department of Physiology and Kinesiology, as well as doing a PhD at SFU and Harvard in Kinesiology and Applied Mathematics, and then did a postdoc in rehabilitation research in the Department of Physical Therapy from UBC, which led her to her current position as a postdoctoral fellow here at the U of C with Dr. Walter Herzog. She didn't leave me an interesting fact, so I'll make one up about her. She's very smart, intellectual person, but she doesn't cheer for the Canucks, so that's good for her. Today's Steph's talk will be changes in skeletal muscle properties and mechanical function following chronic stroke in rats.
Awesome. Thank you very much. It's hard to be a Canucks fan these days, so can't blame me. So thank you everyone for being here. So today in this seminar, I'm going to be kind of following up on a seminar that I gave a little over a year ago now, where I talked about kind of some of the methods and kind of a proposal of the work that I was going to be doing, looking at muscle function changes in rats with stroke. And now we have some kind of preliminary results coming in, so I'm going to be kind of adding to that story and sharing some of that today. So stroke is one of the leading causes of disability globally, and it can cause some pretty substantial mobility challenges. And so stroke is typically results in hemiparesis. And so what that is essentially when you have a stroke or a lesion on one side of the brain, and then you get deficits or paresis, weakness, things like that on the contralateral side of the body. And so studies looking at muscle function in humans, in people with stroke, have found that relative to the non-paretic side or the unaffected side in individuals with stroke, the paretic muscle has lower cross-sectional area and volume, so atrophy, increase in tissue stiffness, and then altered regional activation patterns. And so this figure on the right is a study where they looked at shear wave velocity, which is a proxy measure for tissue stiffness. And you can see here, so here's the non-paretic side of individuals with stroke, and then we have the paretic side, and then age-matched controls. You can see that on this paretic side, there's an increase in this shear wave velocity or this proxy measure of tissue stiffness. So that's kind of showing some of these indirect measures in humans. And so while these muscle changes have been correlated with reduced joint torque, we have very few direct measures showing changes in mechanical output of muscle following stroke. So in humans, we're really limited in terms of what type of measures that we can take. And so we really just can't get any direct information about the muscles in these individuals. And so rat models of stroke can kind of provide us with some more of that direct information on what's happening with the muscle following stroke. And so of the few studies that have looked at rat models of stroke and whole muscle function in those animals, there's this study from Brian McIntosh's group. So they looked at whole muscle function, so the active force length and force frequency properties of the muscle 2 weeks following stroke. And you can see, so the control animals is in this blue color, and you can see that the force kind of maintains or plateaus. And then in the stroke animals on both the paretic and non-paretic side, there's this kind of force decay or decline. So it's not able to essentially maintain tetanic force. So there's that drop in force in those animals. And then another study where they looked at muscle function, whole muscle function 4 weeks following stroke. And so they found some minor reductions in active force in the stroke muscle or the paretic muscle, and then also some increases in passive stiffness. But in comparison to human studies, and a lot of the studies, these indirect measure studies in humans, they have typically looked at chronic stroke. And so that is defined as greater than 1 year post-stroke. And in a lot of these studies, it'll be like on average 8 years post-stroke in these individuals. And so these studies looking at 2 and 4 weeks post-stroke, that's very early. And so that might not kind of reflect the full changes that people are seeing kind of in humans. Yeah, so the time between testing was quite short. And then also, we're not getting any idea of kind of the smaller structural scale. So this is all at the whole muscle level. So it'd be interesting to kind of see across different structural scales if we can kind of tease apart these mechanisms that are leading to some of these whole muscle deficits. So that kind of led to the work that I'm doing in Walter's group. And so the objective the overall objective of kind of my stroke research has been to examine changes in muscle properties and function across structural scales following chronic stroke in rats. And so that's kind of further along. So after the stroke has been induced, waiting a period of time for kind of changes to propagate before testing the muscles. And so for this study, I looked at 28 10-week-old female Sprague-Dawley rats, and then I allocated them to either the stroke or sham groups. And then for the stroke animals, the strokes were induced using a photothrombotic stroke model. So what that essentially is, you can see this image on the right. So the animals were deeply anesthetized, so they were put under during surgery. And then I fixed their skull in a stereotaxic frame, and then removed or opened up the skin on the surface of the skull, kind of scraped away the periosteum or the layer of connective tissue over the skull, and then kept the skull totally intact. And then kind of positioned this light here over the region of the brain that I wanted to induce the stroke in. And then I injected a photosensitive dye into their tail vein, waited a couple minutes for that to circulate into the body, and then turned the light on over the surface of the skull. And then rats have translucent skulls, unlike humans, so the light can penetrate to the brain. And then, so that light penetrated into the region of interest that I wanted to induce a stroke in, interacted with that photosensitive dye, and then damaged the tissue and induced a stroke. And so for the sham group, or the controls, I did the exact same procedures, except I just didn't turn on the light, essentially. And so then in terms of the study timeline, so the animals, they arrived, and then we did some acclimation and training. I did some additional beam testing, which I'll get into in a couple of slides. Just some tests to see their motor impairments. And so we did some training for the animals for a couple of weeks, and then induced the strokes at 12 weeks, and then waited 18 weeks before euthanasia and then harvesting, and then following that has been the tissue testing. So then in terms of confirming the strokes, so it's possible with this procedure that the stroke just doesn't work, like potentially the light doesn't penetrate the skull sufficiently, or I didn't scrape the periosteum enough, that kind of thing. So I needed to be able to confirm that the strokes were actually happening in these animals. So after euthanizing the animals, I took out the brains, and then we did Nissl staining, which stains the cell bodies of the neurons. And so on the left is a stain from one of the animals in the sham stroke group. As you can see here, so this is the surface of the cortex. And you can see that this is totally intact. And we see on both sides of the brain, left and right, it's kind of a nice smooth surface, and everything looks symmetrical on both the left and right sides. And then in this animal where I induce a stroke, in the region associated with where I shone the light, there is this big kind of gaping hole post-mortem. And so that's kind of typical for a lot of the animals in the stroke group. Some of them were a little bit milder than this, but this is kind of a typical, like a representative image from the stroke animals. And you can see it's just on the one side of the brain. So that would result in hemiparesis in the contralateral side of the body. And so for the final numbers for my animals, so I was a little bit worried that none of my strokes were going to work. So I did way more animals in the stroke group than I probably needed to. So I ended up with 7 animals in the sham stroke group, and then I had 21 animals that I induced a stroke with. 5 of them died or had no lesions, and then 16 ended up having survived the duration of the study and ended up having lesions similar to this that were confirmed with this Nissl staining. So then I mentioned the beam training that we did to examine the motor impairments. So what this essentially is, so in addition to showing that there is a stroke or damage induced to the brain, I also wanted to know that this damage to the brain was doing something in terms of mobility. It was changing the way that they were moving, which would probably relate to some deficits at the muscle level. So on the top is kind of an image of what this beam test that we used kind of looked like. So it's called the beam traversal test, or tapered beam test. And so Hoa actually was kind enough to make this setup. Got his saws out and everything. And so it's thicker, this beam is thicker on this end here. And then they have this box here that they want to end up in because there's a bunch of trees on that side. And so we essentially train them so that they run from this end of the beam all the way over to the tree box. And then in order to test their limb-specific impairments, we essentially just see and count how many times each of their limbs fall off the edge of the beam. And so the task gets kind of harder as the beam tapers. And if they've had strokes, they tend to get worse at keeping their feet on the top surface of the beam. And so you can see in terms of the results, so this is the sham group. And so on the vertical axis, we have the missteps, which is the percent of the total number of steps that they took. And so this is the post-surgery, 1 week post-surgery relative to their baseline. So it's kind of how much worse they got essentially, and for each limb. And so you can see across all the limbs, there was very little change. Like on average, it was net zero change for all the limbs in the animals that have the sham stroke. And then in the stroke group, we see something very different where we see for the paretic hind limb, so that's contralateral to the side where I induce a stroke, we have this pretty big jump in missteps post-stroke. And then there's also some additional deficits on the paretic forelimb. The strokes that I induced were quite wide in the anterior and posterior direction, so I got a little bit into the forelimb motor cortex as well as the hind limb, and so that's probably kind of what's attributing or what's causing this increase on the paretic forelimb. And so that kind of also confirms, in addition to the Nissl staining, that the stroke kind of worked and it resulted in these predictable motor impairments that we expected. Okay, so I mentioned that kind of the objective was to measure muscle function across structural scales. And so there's kind of different properties in these different structural scales that could result in functional deficits in terms of like muscle force and things in response to the stroke. And so one of the things I was interested in is looking at the tissue and fiber type properties. So if a muscle has a loss in fibers, but an accumulation of fat or fibrosis, that will change the force output. So if you have a loss of fibers and that's replaced by fat, we would expect there would be a reduction in force. And then there also could be an increase in passive stiffness if there's an accumulation of this fibrosis with a fibrotic extracellular matrix, which is kind of mostly comprised of collagen. And so looking at these, we can kind of hypothesize how the muscle function will change if we see these changes in tissue properties. And so I don't have the results from these yet. We're still in the process of analyzing these stains, but my hypothesis for this will be kind of coming back to that shear wave velocity study, looking at where they, in humans, found that there's an increase in this proxy measure of tissue stiffness on the paretic side. I hypothesize that there may be more fibrosis in the animals, the paretic side of animals with stroke, and then that could potentially kind of increase this passive stiffness at the bone muscle level. So that's kind of a to be determined for next seminar results you'll see. So then in terms of the fiber type properties, so this has been spearheaded by Latif. I asked him this morning how to pronounce his last name, and he told me not even to try. So he's just Latif. But so he's been doing this really cool immunofluorescence staining. And so these are some sample images which look really nice in comparison to some of the other things that we do. There's some kind of cool pictures. But so this is a tibialis anterior sample slide from that muscle on the left, and then the soleus on the right. And so we have these different fiber types that you can see where they stain, the different fiber types stain different colors. And so you can see for the tibialis anterior, it has substantially more type 2B and type 2X. So it's a faster muscle in comparison to the soleus, which is primarily just type 1 and a few of the type 2A. And so for Latif's honors project that he's just wrapped up, he was looking at the tibialis anterior. And then for a summer student project coming up, he's going to be focusing on the soleus and plantaris and looking at if there's differences in paretic and non-paretic muscle in the stroke and control groups. But for the preliminary results for the TA, so he found that there's lower median cross-sectional area and higher median SFI, which is shape factor index. So essentially the higher the shape factor index, the less circular the fibers are. And that was in the stroke animals compared to the shams. And then he also found that there's smaller percentage, there's fewer and fewer percent of the total cross-sectional area that's the type 2 muscle fibers in the tibialis anterior. And interestingly, the side effects were not significant. So there was a difference between the sham and the stroke group, but there was no difference between paretic and non-paretic within the stroke or the controls. And so that potentially could allude to some either systemic effects. So there could be an increase in inflammation or these kind of metabolic processes that lead to some of these fiber type changes in the stroke animals, and it's affecting both sides of the body. Or it could also be that there's some differences in activity or things like that we didn't measure that could be affecting the stroke group differently from the controls. So then in terms of the single fiber mechanics, we have some preliminary results from that. So this these tests were, preliminary data was collected by Nicole, in our group, who's a master's student, and then Faizan continued on with some more of the testing later on. And so for the preliminary results, so we're finding that there's minimal differences in passive stress between the paretic and non-paretic fibers. And so you can see that in the light gray is the non-paretic, and then in the darker gray is the paretic, and we have passive stress here on the vertical axis, and then we have sarcomere length. So that's essentially you stretch the fiber to greater lengths, and then you measure the stress at those different lengths. And so between the paretic and non-paretic side, there's very minimal differences. But then when we look at the active stress, so when the fibers are activated or they're put into an activating solution, then we see that between the paretic and non-paretic, this dark gray on the paretic side is quite a bit lower across all lengths. So for the paretic side, there was a reduction in the active stress. So that's force per unit area. So the minimal differences in passive stress, so that could be a couple of things that are contributing to that. So it could be that if there's a higher stiffness of the extracellular matrix, and then we partially degrade that by the skinning, and then there's potentially also like in muscle fibers from kids with CP, there's like shown that there's a reduction in protein titin, which also contributes to passive force. So if there's a loss in the protein titin, and so a reduction in passive force due to that, but then there's an increase in the ECM stiffness, so an increase in passive force due to that, those kind of balance each other out and lead to like a net zero difference. Or it's possible that just stroke had no effect on titin or ECM, which I'm kind of leaning more towards, and I'll show some protein results in a second that'll kind of explain why I'm leaning in that direction. And then the lower active stress, there could be changes in the function of the fibers of the sarcomeres, or changes in the sarcomere protein content. So because in muscles where we see atrophy or atrophy fibers, we expect there's a reduction in cross-sectional area. But here we're showing that independent of cross-sectional area, there's a reduction in force per unit area. So that kind of alludes to this idea that there might be some reduction in the sarcomere proteins or how the cross bridges are binding and the force they produce. That's kind of leading to those results. And so again, this reduced active stress could be amplifying the force loss if there is the reduced fiber CSA at the muscle. So then in terms of sarcomere and fascicle lengths, so this was work primarily kind of led by Sydney Dorcher, who was an undergraduate honors and summer student that I worked with. And so in order to look at sarcomere length changes, so in paretic muscle in kids with CP, we see that there's this really exaggerated lengthening of sarcomeres in the paretic muscle. And so CP has a similar kind of mechanism of action, like there's a lesion, a white matter lesion in the brain, and that changes innervation of the muscles on the contralateral side. So I thought maybe that this is something that could be also happening in stroke. And so to look at that, essentially Sydney isolated some bundles of fascicles, and then she shined a laser over those fascicles, and then you can get this characteristic diffraction pattern. We just have a piece of white paper underneath, and then you can just take a picture of this diffraction pattern. And then from that, you can measure the median sarcomere lengths, and then also the dispersion, which is a measure of kind of heterogeneity in sarcomere lengths. To kind of see if those similar changes in sarcomere lengths are also happening in stroke, and like the way that they are in cerebral palsy. So kind of from the results, it looks like that's not the case. So there's kind of a lot going on this plot here, but we have sarcomere length on the top, and then we have sarcomere dispersion, or the heterogeneity in sarcomere lengths. And so in the light gray, so these four on the left is the extensor digitorum longus muscle. And then on the right is the lateral gastrocnemius. And then we have our paretic in the hollow circles, and then in the solid is the non-paretic. And then the diagonal lines is the sham, and then the solid is the stroke group. But you don't have to really remember any of that because there's no significant effects other than just muscle effects. So other than there's a significant difference in sarcomere length and dispersion between the EDL and LG. And so that's likely because we kept the muscles on the limb or the legs, and then pinned the joints at 90 degrees for the knee and the ankle. And that results in essentially differences in the force length positions of those muscles. And so it's kind of expected that given those conditions that we see differences in the sarcomere length and dispersion, but then there's no effect of the stroke or or like a paretic or non-paretic versus non-paretic versus, and then also control versus the stroke group. So like I said, there was a significant difference in the length and dispersion between the muscles, probably because they're just on a different position on the force length relationship, which is kind of not that exciting. But these sarcomere results are consistent with some previous work done in humans, and they used indirect measure of microendoscopy to measure sarcomere lengths in humans, individuals with stroke in the bicep brachii muscle. And so on the right here, they have the sarcomere length, and then they have fascicle lengths on the left. And so you can see that between paretic and non-paretic sides in the stroke group, there's no significant difference. But then they did find, which we have data for, but haven't analyzed the results. I haven't analyzed the results of yet. That they found a significant difference in fascicle length and serial sarcomere number in the paretic versus non-paretic groups in the stroke animals in comparison to the control, but then the sarcomere lengths weren't any different. So there's this kind of atrophy effect, but without like this over-exaggerated stretching of sarcomeres that we see in CP. So that kind of may allude to like potentially less deficits in muscles in people with stroke in adults that have these white matter lesions in comparison to kids. So yeah, so that's still to be determined what's going on with the fascicle lengths and if they parallel these results in humans. So then in terms of the protein content, so I mentioned before that I'm thinking that the change in, or the no difference in passive force between, or passive stress between paretic and non-paretic sides in the single fibers is due to potentially just no effect of stroke. These protein results that are from Didon, who's an undergrad who just wrapped up his honors project, kind of support that idea because he found that there's no difference in titin, nebulin, myosin, and there might be some slight differences in actin. They're kind of not significant yet, but potentially with more samples, they're kind of approaching that direction. But there is this difference in total protein content between paretic and non-paretic sides of stroke animals. And so even though there's no differences in the ratio, there's just a reduction in proteins in general. And that kind of alludes to the potential that there's these proteins are being replaced by something else in the muscle. So potentially collagen due to an increase in fibrosis or fat or things like that. And thank you to Venus for sending me these results. Yeah, so no significant difference in specific proteins, but there does seem to be a difference in total protein content. Yeah, and so then in terms of the myofibril mechanics, so those have been collected previously by Taiki and Choulier. And so we're still working on analyzing some of that data. And so this is essentially to measure the passive force of the myofibril in paretic versus non-paretic muscle. And so what we would expect, so at the myofibril level, passive force is due largely to the protein titin. So we don't, at the myofibril level, we don't have any of the collagen or fibrosis or extracellular matrix that's contributing to passive force. So it's just largely or totally due to titin. And so in kids with cerebral palsy, which is kind of where I'm related to, there's a lot more research done in that population. They found that there's a reduction in the content of titin, and then that's associated with a reduction in passive force of the myofibrils. But because we haven't been seeing any changes really in the titin content, I hypothesize that there really isn't going to be much change in the passive force of the myofibril level when we have that data analyzed. So again, that's next seminar results. Okay, so taking this all together, so in summary, so there are some small fiber type changes in the TA that Latif has found so far, and it'll be really interesting to see what happens with the other two muscles, so the soleus, which is primarily a slow muscle, and then the plantaris. And then there's no differences in sarcomere length, so the heterogeneity between sides or groups. There is some reduced active fiber stress, but there's no difference in passive fiber stress in the paretic compared to the non-paretic groups. There is reduced total protein content, which is kind of potentially alluding to the fact that there might be an increase in fibrosis or fat content. And so then to just kind of tie this all together, once we have all the fascicle lengths and then the fat and fibrosis content, and then fiber type changes for other muscles, and then those passive myofibril forces, will be able to kind of link everything together. But something that's been kind of interesting to me and kind of a bit of a takeaway, other than the passive or the active fiber stresses, despite these really like big holes in the muscle and pretty substantial mobility deficits, these animals don't seem to have like very big deficits in the muscle, which I kind of have found a bit surprising. So other than those active fiber stresses, most of the changes have been fairly small so far. So in terms of next steps, so there is a student, Nicole, who I kind of mentioned before. She's doing some work kind of bridging or extending off of this work, looking at the effects of menopause on post-stroke changes in single fiber mechanics. So we saw that there's that change in active force or active stress in the paretic muscle fibers. And postmenopausal women have worse mobility outcomes following stroke in comparison to younger women and age-matched men. And so we were kind of interested in knowing if that is due to differences in biomechanics. Like, is there some mechanism, for example, estrogen, that's could potentially amplify those reductions in active stress that we see in the paretic muscle in stroke animals? And so for this study, she has 4 different groups, and she's doing, this is just kind of a pitch to come see her future seminar. But we have 4 groups, or Nicole has 4 groups, where she induces menopause with ovarectomy. And then she also has similar structure with the stroke and sham stroke groups, as I had for my study. And then preliminary results, she's done the full study, had 40-something animals, and she's just kind of looking at some of the data and then she's collecting some of the single fiber experiments. But her preliminary results from her uterus mass results to confirm the ovarectomy have shown that that's been pretty successful. We have this pretty substantial reduction in uterus mass in the animals that have had the ovarectomy in comparison to the sham. So that's kind of providing some support that this is like a procedure that's working. And so stay tuned for that. And then other potential future directions. So again, we're already kind of looking at some of the menopause stuff, but looking at additionally the impact of aging. My animals were very young, 10 to 12 weeks when the stroke was induced. Is possible humans that have stroke tend to be quite a bit older. And so it's possible that that difference in age at onset of stroke could be influencing the magnitude of the effects of on the muscles. And then also a lot of individuals with stroke, people with stroke have a lot of comorbidities. So things like type 2 diabetes, cardiovascular disease, that could also amplify these post-stroke changes in muscle function. So kind of exploring some of those will give us a fuller or bigger picture of what's going on in terms of muscle changes following stroke. And then also looking at time courses. So I mentioned kind of this idea of chronic versus acute. We really don't know when those changes are happening. If there are is there some kind of threshold where this is happening? Does it all happen within 2 weeks? Is it, we just have no idea of what that trajectory looks like. So kind of future work can kind of look at how those kind of changes occur over time and then when interventions could occur. And then also now that we have this model kind of established, we can use it to explore the effects of different interventions. So things like Botox, where those have primarily been evaluated on healthy muscle, but now we could potentially look at if the effects of things like Botox are different in paretic muscle in comparison to unaffected muscle. And then also another area of interest, I would be interested in looking at what are the effects of stroke on whole muscle dynamic function in vivo. So we can use these kind of different measures. So this is fluoromicrometry, where we use these like orthogonally placed X-ray arms to look at time varying muscle deformations during gait in live animals. And so seeing how stroke affects dynamic muscle function would be pretty cool as well. And so this has really been a kind of group undertaking, as you've probably seen from some of my slides, and a lot of people have contributed to it. So thank you to everyone who's contributed, and also thank you to all of you for being here and for the funding sources as well. Thanks.
All right, thanks, Steph. So before open for questions, let's get started.
For the immunofluorescence staining, what's the target structure that differentiates between muscle fiber types?
So there is the myosin heavy chain isoform, or like the myosin protein. And then there's antibodies that bind to those particular isoforms of the myosin. And then they have different colored like immunofluorescent tags. And so then when they're put under if it's a particular isoform of myosin, then a particular antibody with a particular color binds to it. And then when it's put under light, then they show up as different colors. Is essentially how it works.
So it's a myosin
Yes,
structural differences.
yes.
I'm just curious about your lesions you're doing.
Yep.
You said they're fairly consistent across, like generally fairly
Ish.
same-ish, okay.
So I
How
would
much
say
control do you have over the size and the sort of depth and?
less control than I would have guessed at this point. It might get better like with the more I do, the more, but it didn't seem to be the case. Like over the course of the animals I did, it wasn't super predictable, even though I kind of, it was pretty consistent in terms of how, like, I was placing things. Yeah, some of them for some reason just don't have, some of them have quite mild strokes where you just see a little tiny dip at the top. And then others have it quite deep where it's like into subcortical regions. And there doesn't seem to be like a clear reason why that's happening. But then that also has been the case. So Dale Corbett, who has done, years and years of these kind of studies, they also find that some animals just don't have strokes, have unsuccessful strokes. And there's no,
Yeah, it's neat, because I mean, just speaking with Tyler, he'll say like, he'll have people come in with a massive lesion, have no effect on their upper limb.
yeah,
Motor control, like kind of base set.
yeah,
Or they'll have like a tiny lesion just at the right spot and be entirely compared.
Yeah.
So it's neat.
so I
It's good
think,
you have some variability
yeah.
in the lesions you're getting.
I would say, so they're all generally in the hindland motor cortex, but, yeah, it's also interesting, doesn't seem to be, I looked at the correlation between the beam results, like the motor impairments, and this like approximate severity of the stroke, and there didn't seem to be like a very strong relationship there either. Yeah, so it's not really clear. So it doesn't seem to be like directly related to like the size, yeah.
Thanks, Steph. It's very nice presentation. I have a question about the level of activity of the animals.
Yes.
So, is there like a huge difference?
We don't know because we didn't directly measure it, but the interesting thing about rats and why I'm potentially thinking it might be good to consider or explore other animals is because they bounce back surprisingly fast after the stroke in comparison to humans. And they have very little obvious deficits when you just watch them walk. Like just watching the animal, if I just put the animal on the beam or on a table, I wouldn't be able to discern which one has a stroke and which one doesn't. Whereas it's very apparent generally in humans. And they like start walking as soon as they recover from the anesthesia. Like there's no really period where they're totally unable to mobilize. Whereas that's not really the case in humans. Like remobilizing takes a bit longer. What was your question? Yeah, so my guess is that there isn't substantial, they might be a bit slower to get back up. And start, like their activity level might be a bit slower in the beginning, but I wouldn't guess that there would be a huge difference in the animals. But yeah, we have to actually measure that. In order to be able to tell. But I will also say that, so these animals don't have wheels. So they're not able to really substand, they're double housed, they're double caged. And so there isn't a whole lot of room for them to move, so I wouldn't expect that there's like huge differences between the shaman, just because they all can't really move much. So, yeah.
We have an online question for you. So corrected muscles, fiber is smaller in cross-sectional area. So was the whole muscle group of the cross-sectional area of muscle volume are also smaller in paretic group compared to non-paretic group?
So, like from the histology probably?
Yeah.
So Letitia did look at that, and I
Okay.
think that there was a difference, but I think it was the control were smaller than the stroke, but I think we can't really determine from the histology because there can be regions that are lost from the cross sections. Like there could be kind of chipped areas on the edges and things like that, that make it so that we're not actually measuring. Like I wouldn't be super confident in those results.
Because somebody said in the Atkins paper, the mean sacromial length seemed to be about 3.5 micrometers. which is long, so he wonder how they measure sacral length.
Yes. So this is from the microendoscopy, but I think it was second harmonic generation, maybe Anquan would know more about that technique. But I don't know the details of how exactly they, but yes, that is quite long. Like mine were 2 point, more in a typical range that you would expect, whereas like 2.4. Like this is more kind of like a average sarcomere length or like a normal sarcomere length, whereas these are quite long, even though so this is kind of more in the range that we would expect, I think, from kids with CP. They're very long lengths, but in kids with CP, you expect there's a big difference between paretic and non-paretic that they don't see in this study. So I don't know if that very long length is an artifact of the measurement technique, or if that's because these individuals, but my guess is that it's a measurement technique artifact, because the individuals with no stroke also had that, but it also might be a muscle difference, like bicep, or where they were on the operating line. Like if they had the elbow fully extended, then the muscles could just have
Okay.
long sarcomere lengths.
Okay.
How does your stroke model differ from the way humans get strokes, or is it the same, damage is same, or?
It is different, yeah. So that is one potential limitation. There's different models of inducing stroke. There's things like endothelin 1 injection where they like inject it into a particular region. There's also middle cerebral artery occlusion where you like block a blood vessel, and that probably is more closely related to like, or directly translates to an ischemic stroke. But that's a very difficult procedure and like there's a high mortality rate essentially with that, and it's can take a really long time to get like proficient in that procedure. And they also tend to have a lot of other issues that you don't want, like subarachnoid hemorrhage and all those other things in those animals. But yeah, so in humans, most strokes are ischemic. So it's like things like an atherosclerotic plaque or like narrowing of the blood vessel or a blood clot or something like that blocks blood flow and oxygen being received by the brain tissue, and then that tissue in that area where the blood flow is going dies. Whereas here we're like damaging it directly due to the light. And so the region would be very different. And then also potentially the like inflammatory cascade that results afterwards would also be different potentially. So we just like immediately kill off the cells where like if there's ischemia, there's like a different kind of cascade of events that happen afterwards. So yeah, that's potential limitation. It's like the regional, there's like regional differences or like spatial distribution differences because of different mechanism, but then also like this inflammatory cascade might also be contributing. Yeah.
Thank you, Steph. About your speculation about why do you think that the stroke did a really minor function affection in this ranch?
I don't know. Long story short, but there's a bunch of factors. So I said the age thing, so that's potentially one. So in a lot of the studies in people with stroke, they look at people who are 60s, 70s, 80s. And then there's also a bunch of factors that it might be like an animal, like a species difference. Rats are very resilient, like they're kind of gross sewer animals that don't get infections. Like they're hard to kill and they bounce back so fast. And humans are not like that. Humans are way less resilient. And so there might be something like their immune systems are so robust and they have these different inflammatory processes that they just don't have the same deficits in response to stroke as humans. It could also be differences in how they're, quadrupeds versus bipeds. It could be differences like comorbid diseases in humans with stroke, lifestyle factors, differences in like rehabilitation and even medications. Like there's some kind of emerging studies that like the medications that they give following stroke and like blood pressure medications and things like that can affect muscle. So it's possible that some of the interventions that we give to people with stroke that are beneficial for them not having brain problems or more strokes could propagate deficits potentially. So yeah, there's a lot of potential reasons why it seems to be different. I have been kind of surprised that things are as mild in these animals in comparison to like when I did testing in people. Like you see a huge differences on the paretic versus non-paretic side.
have you seen the same stroke model in other quadrupedal animals? Or,
I think I've seen some, I think it was sheep that maybe I saw some. There is some other animals, but the problem, especially with the photothrombotic model, is that rodents like mice and rats, it's fairly straightforward because they have translucent skulls, but other animals, you have to like shave down their skulls to make it translucent enough. So then and then also bigger animals are kind of difficult. And if you think of a rabbit, then they hop, so that's not super great translation to humans. I was thinking something like mini pigs, would maybe be it, because they're like, they don't hop, they're bipedal, but they maybe aren't as resilient.
Hmm.
And they don't have all the problems, like, they're not so sensitive as rodents.
When it comes to the muscle wasting that you see post-stroke, what's the prevailing hypothesis about? Not hypothesis, but like, what do people think is primarily responsible for these changes? Is it mechanical loading, or is it some kind of neurogenic factor associated with the stroke? What do people think it is?
Well, so I think in general, like in stroke recovery research, people primarily just think it's due to changes in innervation, like due to, like they consider it like motor recovery. They're not really at all considering what's happening downstream with the muscle.
Yeah.
But I like kind of what I was thinking that it would be similar to like de-innervation studies,
Okay.
where like you cut off the innervation and then
Yeah.
you get this like massive, or like immobilization studies where you get this like massive atrophy. But then I would also guess that there are some like systemic inflammation kind of stuff that's in there. Yeah.
Just about your technique to induce a stroke.
Yeah.
I guess it depends very much on the stresses of your brain, right? How
Skull, yeah,
on the scalp,
yeah.
brain. Yes.
Brain also, I guess, yeah, would affect the depth.
So the focus point you want to induce lesion to, is it in the middle of the brain or on the surface of the brain, or?
So so I go from the surface, so it's going from surface to deep. And then I position the light based on the anatomical landmarks on the skull. So there's like, the sutures on the surface of the skull, like the lines. And so I do it like a particular distance from like the bright line and then the interhemispheric line suture. And that's how I position it.
So when you do the scanning to confirm the location of the lesions.
Yeah.
They normally land at a similar locations in the brain, or?
So Ruth and I had a lot of discussions about this, and then we ended up just taking kind of, my intention at this point was just to confirm that there was a stroke, as opposed to quantifying details about the magnitude and specific location, because I think in order to do that well, we would need like MRI, kind of, because there's limitations in terms of like the slicing with the histology, getting that, so it's like very clear where the position is. So I don't know if it's exactly the same. Yeah.
I'm just wondering if, if you look at the scan sections, right, the locations of it, and you compare it to see if there's a direct correlation between the mobility loss.
Yeah.
And then from there, you can then tease out get the function loss. To see a more drastic change than the one that you are.
Yeah, so that is something that would be cool. I need to get a rat MRI to scan the veins. But that would be really cool and also to look at, like, people will just be like muscle function. But my guess is that there's, depending on the specific region that you induce a stroke, there could be differences in innervation and effects across the body, like differences between flexors versus extensors, differences, in that respect. So I think that would be really cool to be able to like link those things, which hasn't really been explored.
Doesn't Jeffrey Dunn have a MR, big fancy MR that he uses on?
I don't know, doesn't.
Yeah, it's like 7 Tesla or 5. He's putting it
Yeah.
on a human. Like, I feel like it would make a human sick. I think it only is used preclinically.
Okay.
James?
Next up step. Could you find your hypothesis about the PCM and that it was not changing in single fibers? Cause I was a bit confused there.
Okay, so the passive force didn't, okay, so my original hypothesis back before the protein results was that there would be an increase in the ECM content, potentially stiffness as well. And then in the skin, and then there would be, like, this is basically from CP, the kids with CP results. And then there would be a reduction in titin. And then the ECM is partially degraded. And so that combination, the passive force would be balanced out, and then there'd be no difference between phoretic and non-phoretic. It's like a titan loss, but an ECM increase, so then net zero. But since the protein results, where there's a no difference in titan so far, I'm hypothesizing that there's probably no difference in like ECM stiffness, or it's like, and there might be a slight difference, but it's kind of not really, it's like negligible or canceled out by the skinning. And then there's no difference in titan, so then that's why the passive force is not different with the single fibers.
Okay,
Is that
so
clear?
yeah, I agree with the skinning canceling out the effect of ECM. But ECM being higher in the phonetic muscle, I think the protein dissolve will support this hypothesis.
Yes. So yeah.
It seems that there's a decrease in the protein very muscle mass, so it seems that likely there's an increase in collagen and fat.
Do you think it's potential? Yeah.
So I would,
But based on the no difference in passive force at the single fiber level, but I guess at the single fiber level, it would still be primarily due to titan, not ECM, even though there is some there, but it's not. And it's mostly degraded anyway, so.
maybe we did a good job with the skinning process and it's all.
Yeah, it's all gone anyway. Yeah, that's possible. That's another option. Yeah. Or I guess there's potential that it could be mostly a change in fat. And that's what's leading to the change in protein content. But I haven't really seen, just looking at the slides, it doesn't look like there's much fat in anything. So, yeah, they'll be interested to see.
Then have you considered to take a look at this final level activity, other units? Because that connection that possibly, that you were saying, argue for people that rehabilitate this patient, think that is more like a activity dysfunction.
Yeah.
How the muscle is activated, so they attribute the changes of the activity of the brain towards the spine, no?
So that was something I thought about early on, but I put that in like a future directions bucket. But I think it is interesting because I wanted to measure spasticity.
Mm.
But most of the studies that have looked at spasticity in rats, like they don't seem to have much spasticity in comparison to humans. And so that also could be the difference between, that's contributing to less severe differences is the lack of spasticity. And because I think there's some hypotheses out there that spasticity is what contributes to, like in kids with CP, that it causes a lot of issues in phoretic muscle. So yeah, so it would be interesting to see if there is changes in spinal activation. Yeah.
Follow up, like the rewiring of the brain and so the muscle, like the neuroplasticity.
Yeah.
And so the muscles are not as denervated as you think they are.
Yeah, and it's also, so I guess I've always thought that, the muscle changes would get more severe over time. But that might not necessarily be the case. If you have like reinnervation, and like neuroplasticity where other regions are able to like take over, other like dendritic projections can like take over some of the function of the lost neurons. Then the deficits might, like there could be an initial loss of deficits and then plateau, or there could be some like recovery of function potentially. Yeah, it's hard to say. That would be cool.
Does red stroke by itself spontaneously?
Say that again?
Does red go on a stroke spontaneously? Or
Rats have strokes.
Rats have strokes. Oh, I have no idea. Yeah, that's a good question. We never thought about it. My guess is yes. Does anybody know? I would guess that they can get like blood clots and cardiovascular disease, like maybe in the diet-induced obesity rats that we get some strokes eventually.
Or maybe natural selection just keep it around.
Yeah,
Then the one they tested, they are the most resistant one. That's why they don't show them.
yeah,
Yeah.
yeah, it's very interesting how resilient they are, so maybe these are like the super rats that survive generations of sewage.
And green ooze.
Start with like this question, so we saw that it's like I'm looking at the same complex. The first order diffraction seems pretty wide.
Yeah.
And it's likely because you're measuring the software lines not in one fiber.
Yeah.
It's these are bundles, right? Okay. So it could be just differences between fibers. That's not softwares in series having different software lines.
That
You
is
have
potential, yeah.
fibers in your bundle.
Yeah, and I was also trying to think about like the offset, like you have bundles that are not like, there's not like they're perfectly aligned like this. And if you have, they're in parallel and they're all offset, like how would that affect that dispersion? So yes, I need to think more about that and like the implications and limitations of that. But yeah, there's no really difference anyway, so.
Do you know if there's any change in the ventilation angle of the metal? If this could change also.
I don't know about-
about.
Yeah, I don't know about these animals, we haven't looked at like architectural properties. But I think in humans with stroke like measured from MRI, I think, maybe also ultrasound, resting architecture is always tricky though. But yeah, I think there's a reduction in initial like resting pinch angle with atrophy. Reduction in initial pinch angle or like, and then also a reduction in rotation, vascular rotation with contraction in paretic compared to non-paretic.
Does this disagree with the fibers being or the maximum being short?
Not necessarily, because you could get a reduction in. Yeah, not necessarily, like it would, you could get a decrease in thickness from decrease in vascular length or a decrease in pinch angle. But those are not necessarily, yeah, that doesn't necessarily translate to vascular length with pinch angle.
You know if the type of collagen is the same in rats as in humans? And like, would that influence the difference in like spasticity that you would see?
I'm trying to think, I don't remember. I was just reading about after, like, collagen types and amaurosis. And I don't remember seeing if there was, I feel like Walter's probably watching this and knows the answer to this. But yeah, I don't know. Okay, yeah.
It's type one, I believe it's type one.
I think it's primarily type one, but I don't know if
One,
there's differences in both animals. There's no,
I think so.
okay. I think so.
Any more questions? Okay, then we'll thank Steph for her talk.